what I'm going to talk about you is the methods that we develop in our lab in analysis of the networks. So Dr. Kanuri Rao has set the stage very nicely. He has shown that network biology, of course, is very important. What I will try to show you is that how do we develop methods in analysis of biological networks. I'm going to give one example in somewhat detail. Initially, a couple of examples I will just skip through. So what Kanuri Rao showed us is that there are two approaches, traditional approaches in biology. Rather, one traditional approach in biology, you want to study any particular system, we break the system into smaller and smaller components. So for example, you want to study biosphere, we break biosphere into smaller components such as different populations, and the inferences obtained by studying different populations, we generalize them for the biosphere. Or if you want to study the populations, we study different organisms in the population, and whatever organisms we understand, from there we generalize it to the populations, and so on and so forth. So for example, you want to understand cells, we try to break the cells into different molecules, study the molecules in the cells, and then try to understand how cells themselves behave by trying to understand the molecules. Now, this is a very traditional approach in biology, followed since, I would say, since the advent of the molecular biology in the 1940s and 1950s, and what we call as reductionism. Right? So this is a typical reductionism approach in which a complex problem is broken into smaller and smaller problems. You try to understand smaller problems because they are easier to understand, and once you understand a smaller, you gain sufficient understanding of the smaller problem, you try to generalize to the next level, in whichever way this can actually happen. So this was essentially proposed, propagated by physicists who said that nature can be understood by attempting to understand the parts of the nature. Typical physics problems are actually done this way. Right? I mean, molecules and atoms and subatomic particles and things like that. On the other hand, there has been a growing realism that we can't understand biology by breaking into smaller components. We have to actually understand biology into a whole, what Kanuri Rao very beautifully demonstrated, and that approach is called as a holism. And this is actually what we are driving towards in the last few years. I would say last about 10 years, the realization has been somewhat more stronger that we have to understand biology in its holistic point of view. And for example, it has given rise to certain very interesting hypotheses. For example, how does individual behavior aggregate into collective behavior? So in a population, for example, in this auditorium, if all of us start clapping, you would realize that we start clapping at different point of times with different frequencies, but within a few seconds, there will be synchrony which will emerge. So let us do this experiment. Let us all start clapping. Let's start. Did you realize that within a couple of seconds, the synchrony emerged, right? And that synchrony emerges automatically, what Kanuri called as the emerging property, right? So how does individual behavior aggregate to give a collective behavior? Individuals don't kill each other normally, but if you listen to a very fiery political speech, the amount of hatred that it generates, you can actually start, even a reasonable human being can pick up a weapon and kill another human being, right? And that's a very peculiar way that actually happens. So there is a disconnect between individual behavior and aggregate behavior, and that's exactly what holism approach tries to address, that how does this actually happen, right? So what we have practiced biology in a reductionist way does not give you complete understanding of biology, and therefore we must try to understand it more holistically. So the hypothesis is that to understand a complex system, such as any of the biological system, each of these is actually a complex system, one must understand interactions among different components in the systems and see how the emergent properties arise, right? And that's exactly what Kanuri Rao gave beautiful examples from his work in this particular thing. What I'm going to highlight are actually some of the things that we are trying to develop in our lab as methods in trying to understand this, right? And I'm going to give you one major example toward the later half of my talk, but a couple of small examples to begin my talk. So this is what Dr. Kanuri Rao said, that any complex system is made up of many different small components, and these components interact with each other, right? And what he showed was a protein-protein interaction network. There are many different proteins in a cell. All of them interact with each other with different affinity, with different strength. Not every protein interacts necessarily with every other protein, right? There are very selective interactions, but the selective interactions of these are the ones which eventually will give you a biological property. So a phenotypic property of a cell would come from the interactions between the proteins in a cell, right? How a cell responds to an environment or a change in environment or a mutation all that would actually come up because the interactions in the cells actually change. The protein-protein interaction in the cells change, and they would actually give us the phenotypic property of a cell. And the same thing with multicellular organism, it would happen to the organism, or in many different organisms, it would happen to a population, and so on and so forth. So we must understand how these interactions are actually taking place and how they change in response to certain stimuli. Alright, so that's the thing that we do. Now, to understand a network, we a very simple mathematical formalism that we use. All the networks, we reduce them to matrices. For example, if this is a network of 5 different entities, what I have called as V1, V2, V3, V4 and V5. We can, for example, what we call them here are vertices. So the 5 different entities in a protein-protein interaction network, this could be 5 different proteins. In such a case, protein 1 interacts with 2 and 4, but not with 3 and 5. Protein 2 interacts with 1 and 3 and 5, but not with 4. And the same thing can now be reduced to a matrix form, that V1 interacts with 2 and 4, but not with 3 and 5. So you can actually write it in a very simple matrix format, and you can see that matrix is symmetric across the diagonal. Alright, so the advantage of writing in a matrix format is that we can actually perform many mathematical operations very quickly. And some of the mathematical operations, we'll see what kind of mathematical operations we can perform on them. Now you'll realize that in this case, the flow of information is not necessarily from V1 to V2 or V2 to V1, but we have actually not indicated any direction of the flow of information. Now, biological systems is very common that there is a flow in a particular direction. What Dr. Kanirao showed in the metabolic network, there are different metabolites in a network, and the flow of information was actually going in one particular direction. Right? If you start with glucose and end up with, let us say, ATP, you go through a series of reactions, but they were going in one particular direction. You don't necessarily go from ATP to glucose. Right? I mean, that generally, it's in principle, all reactions are reversible, but in normal, if you feed ATP to a cell, you don't start getting glucose as an output, right? So basically the flow of information can be represented by these arrows. For example, in this case, V1 can regulate itself, or V1 can regulate V2, or V1 can regulate V4, typically in a transcription factor network. Then you can actually represent that once again in a similar matrix. But this matrix is no more symmetric across the diagonal, very similar thing. And in more general form, the interactions between different vertices are also weighted. The amount of information that can flow between different vertices can be less or can be more, and that also can be represented in the matrix. The advantage of representing this in matrices is that matrix operations are very easy to perform, and we can actually start now asking some very elegant biological questions using these matrices. Let us see how we can do some of them. So just to give you a popular example, this is a figure that all of you are aware of. Those of you who are bored would be already doing this. You can actually try to draw this figure without lifting your pencil or without lifting the pen on a piece of paper, and ask a question, can you draw this figure without lifting a pen or paper on a paper? Right? It's called as a Boyd puzzle, and we know that answer is no, but answer is no can be proven very elegantly by reducing this to the matrix format what I showed you earlier. For example, this can be considered as 4 nodes here, or 4 vertices at the corners of the square. And these 4 nodes are connected to each other by the connections which are shown here. And then eventually you can come up, we can see that each of these nodes has odd number of connections, right? This has 5 number of connections. This one has 5 connections. This has 5 connections, this has 5 connections. And to go from any node to another and to exit from any node to another, you need even number of connections, right? However, in this case, each of the nodes has odd number of connections, and therefore this figure cannot be drawn without lifting the pen. It's very simple. It was actually originally stated in the 18th century, what was called as the problem of 7 bridges. Right? I'm not going to go into the problem of 7 bridges, but I mean, that's the origin of that particular problem. But the networks being so complex, very often we are encountered with the questions that are the two networks similar? Are they the same? Right? And this problem was posed to the mathematicians. And about a year and a half ago, this was solved in University of Chicago, what is called as a graph isomorphism. These two graphs, we can't tell by human eye whether they are same or not. But if you do mathematical operations on them, the matrix kind of operations what I showed you earlier, they are actually identical. These two networks are identical to each other. Right? I mean, if you look at the color codes, the blue one is connected to a green and yellow and white. And if you see here, it is connected to green, yellow and white, something like that. Alright, so this is something to do about that. So we come back to biological systems, and start with the hypothesis that we should understand networks and their temporal dynamics and what we can actually understand from that. So we started working on this about 10 years ago. We got interested in the network, in general network biology, and developing methods to understand the biological insight using the networks about 10 years ago. And I will just briefly describe in a couple of slides what we did here. Basically, our problem that we are trying to understand is that if you take a cell and subject it to UV stress, and you take a mutant which does not survive UV stress, and subject that to two different conditions, normal condition and subject to UV stress. So you have basically four conditions: a wild type, a normal condition, a wild type under UV stress, a mutant, a normal condition, and mutant in UV stress. And what are the changes in the networks that you are seeing, right? And whether from there you can understand the change in network correlated to the survival ability of the cell to UV stress or something like that. So for that, we actually came up with this small hypothesis is that in networks, and of course this is not something new, what we were doing, it is already well known, but we are trying to actually apply to a real life problem in this case. So this network, if you take, networks have a very beautiful property called robustness against random node failure. Alright. So in this network, if you see, most nodes what you see here, most vertices have very small number of connections. Right? They have only one connections each. There are only few nodes which have large number of connections. Right? And general natural networks in nature are very similar in property like this. So electricity distribution in the city, for example in Guwahati, or water distribution in, let us say, Guwahati, are networks which are very similar to this. Most nodes have small number of connections and few nodes have large number of connections. There is a centralized place where a lot of information will go out from there, but in most of the places, peripheral places, they would be connected to only one or two. And that is because it's what is called as the robustness against random node failure. And let us try to do this here. In this case, we will try to see how random nodes, when they fail, what effect does it have on the network. For example, if this node fails, what happens is that the overall network, the topology does not change very much. If this node fails, the topology does not change very much. If this node fails, the overall topology does not change very much. So the probability that any node would fail is very high for the nodes which have small number of connections, simply because statistically it is large number of nodes which have very small number of connections. Right? I mean, there's simple probability that if any node would fail, it would be these nodes, they would fail with a higher probability. The probability that nodes such as this would fail is very low because they are very low, small in number. And if they actually fail, you can see that the entire topology of the network changes quite dramatically in such a case. Right? And that's actually is naturally built in natural networks, that large number of nodes have smaller connections, few number of nodes have larger connections, and that gives you robustness against random node failure. How do we apply these to biological networks? Right? Now the property that I described to you is what is called as the degree centrality. So the number of connections to every node is called the degree. The larger the degree, the smaller number of nodes, and the probability of its failure will be lesser, smaller. So nodes which have larger number of degree, they tend to control the network, is what is called as the degree centrality. And it can be very elegantly formulated in a very simple mathematical formula, something like this. But similarly, there are other centrality measures in graph theory, such as closeness or betweenness. For example, if you take all nodes in the network and calculate shortest paths between them, in how many smallest number of jumps that you can go from any node to any other node. So you do a collection of all those shortest paths, and you find that one particular node actually exists on many different shortest paths. Right? So you have a collection of all the shortest paths in a network, but a few nodes actually are recurringly occur on these shortest paths. So these nodes also actually tend to control the overall topology of the network, and it is called as the betweenness centrality. Right? And this can be applied to biological networks very easily. What we showed is that the essential genes, in this case E. coli, so if you take all the genes in E. coli and construct a genome-wide gene-gene interaction network, or the functional network what Dr. Kanir Rao said. So you have 4,000 genes in E. coli. So construct all possible gene-gene interaction network. Let's say you either predict or derive experimentally. And in that network, you apply the three centrality problems that I showed you in the last slide. The degree centrality, the nodes which have very high degree, or the closeness centrality, the nodes which are closer to the center of the network, or betweenness centrality, the nodes which occur on many different shortest paths. What you see is that essential genes, they follow a distribution something like this. the genes which are essential for survival of E. coli. The genes which are not essential or the non-essential for the survival of E. coli, they follow this kind of a distribution. So they have exactly complementary distribution in both of these. Similarly for closeness centrality, similarly for betweenness centrality. So what we can do now is we can actually make a combination of all of these and arrive at, if we did not know which genes are essential for the survival of organism, we can actually, from a network, we can try to predict that these genes are essential for the survival of an organism. Now we can do this, come back to the biological problem that I stated, that wild type subjected to UV stress and Lexay mutant of E. coli subjected to UV stress. So what we have now are the 4 networks: wild type, untreated, UV treated wild type, untreated mutant. This is a Lexay mutant. The gene Lexay makes it susceptible to UV radiation, and UV treated Lexay mutant. And for all of it, we have actually the network with many different nodes and many different connections, what we call as edges. So the connection between different nodes are called edges, and the nodes themselves, the genes or proteins themselves, would be called the nodes. And you actually basically have the overall global properties of all these 4 networks. And then we map it based on the microarray data, which genes are expressed and which genes are not expressed in each of these 4 conditions. So you have a parent network, which is a collection of all the interactions. Then using microarray, you try to address which genes were expressed. So all the genes which were not expressed, you remove them from the network. Right? So you have now 4 networks, each representing every separate experimental condition. Right? So when you do that, the microarray thing shows very nicely that, for example, when you treat the Lexay mutant to E. coli, to UV stress, it turns out that some of the nodes are actually not expressed under that particular condition, under UV. And therefore, they disconnect certain islands from here to here to here to here. These actually nodes are now not connected to each other. So the genes which were earlier interacting under normal conditions do not interact anymore. And that actually breaks several paths of communication between different genes, and that can actually disrupt the overall communication paths between either functions of the genes or proteins or whatever. Right? So that way we can actually hypothesize that these genes were actually therefore important for survival of E. coli under UV stress. Something like that, we can actually arrive at something like this. Okay, there's one example that I wanted to give you. The second example that I want to give you is that, are there motifs in the network which are preferred in nature? Does nature prefer any kinds of motif? This is only a theoretical interest. And what we see is that, for example, once again taken from E. coli, you have large number of transcription factors, and all these transcription factors control expression of certain genes here. For example, RPOS, which is a sigma factor, controls expression of many different genes. Alright. On the other hand, LRP transcription factor controls expression of only one particular gene. So these are all directed networks. As you can see from the arrows, information is flowing from one direction to another. Now in this case, are there any particular kind of structural motifs that are enriched in many different conditions? So basically what we do is that we look at motifs consisting of 3 nodes or 4 nodes and ask question that under many different environmental conditions, by using very similar trick that what we did earlier, take a microarray data, map it on the complete network, the genes which are not expressed, you remove it from the network, and then look for the motifs of such networks and then see what actually you find. And what actually turns out is that, for example, motifs such as this are highly enriched. In this case, the 2 transcription factors interact with each other, but both of them control overall lipid metabolism. So this kind of motif actually is highly enriched. Similarly, what is called as the feed forward motif, one transcription factor controls expression of another transcription factor. This transcription factor controls expression of a gene, but this transcription factor also controls expression of this gene. This is called as the feed forward. Alright, and this kind of motif once again is highly enriched in all the networks that what we saw actually. So we have actually reconstructed here from 500 different microarray data sets mapped onto the E. coli genome-wide protein-protein interaction. We have 500 different networks, and then we are asking a question, which kind of motifs are enriched, and we find that actually these kind of motifs are highly enriched in all the 500 different networks. Certain motifs such as this are never detected. Now this is a feedback motif. Transcription factor A controls expression of transcription factor B. Transcription factor B controls expression of transcription factor C. Transcription factor C controls expression of transcription factor A. So this is a feedback, and this motif is never observed in nature. So the feedback control in the transcription regulator network is almost never observed, right? Because that would actually give rise to a continuous loop, and therefore different transcription factors don't control each other's expression in a feedback manner, something like this, right? I mean, that's turns out to be at least true in E. coli, something like this. So this is the kind of thing that we actually get. It's only a very cosmetic interest, but it's something of something interest to note. Now, the example that I want to dwell upon slightly in more detail, the biological example, is a very fundamental question. The question is like this. If you have a microarray data, okay, in a microarray data, you get a large number of genes which are overexpressed or repressed, right? You are comparing two different conditions, right? When you compare two different conditions, the large number of genes which are overexpressed and large number of genes which are repressed, right? I mean, typical macro representation you would get in green and red dots. That's the representation that we actually make. Now, which transcription factors control this differential gene regulation? That's a fundamental biological problem. Can you pinpoint that a set of transcription factors controls this differential regulation of genes? Alright, that's the problem. So to do that, first we look at how the transcription regulatory network would look like. So simplistically, if you have a gene which is represented in blue and a transcription factor which is represented in red, it controls expression of this particular gene, and we will show in the network as information flowing from red to blue in an arrow. So typically what you would have is something like this, a transcription factor controlling expression of this gene, right? In a network we would show it like this. So you have one transcription factor controlling expression of one gene, or more likely many transcription factors controlling expression of many different genes, or a much more complex thing, that a large number of transcription factors cross-regulating expression of many different genes. And that's a more biologically relevant situation, that you have large number of transcription factors, many of them have cross-regulation of many different genes, and to add to the complexity, certain transcription factors control their own expression, but they also control expression of other transcription factors, right? I mean, that's the realistic biological situation. Now, in a microarray experiment, as I said, going back to that, you have information only from here. What you measure in the lab in a RNA-seq or a microarray experiment is only the expression levels of different genes. And the information going from here to here, that is which transcription factors are regulating the differential gene expression at this level, is lost, right? So the fundamental problem is that from a network, how do you go from here to here? Now this problem is very important because although you find about 1000 genes whose expression has changed between two different conditions, the total control of these 1000 genes is limited only to about 15 to 20 transcription factors, right? And these 15 to 20 transcription factors control the entire expression of all these 1000 genes here. So we would like to address a question, which are these 25 transcription factors? So this is a typical experiment that what you do. So what we do in such a case is that a simple thing. So if this is all the genes in an experiment, in an organism, let us say E. coli has 4000 genes. This represents all the set of all the genes. The differentially regulated genes are represented in this purple box. So if let us say out of 4000, 1000 genes are differentially regulated. And suppose there is a transcription factor X, it is known to regulate expression of certain kinds of genes, that is represented in blue. So there is an overlap between blue and purple. So the question that we ask is that, how significant is this overlap? For a differential gene expression, for example, in this purple circle, how significant is this overlap? And that overlap can be calculated by performing things like a Fisher's exact test, right? I mean, you do a Fisher's exact test, that what was the expected number of genes that were expected to be found here, which were not found, that is here, which were found, and what is the total number of set. Based on this, you can perform Fisher's exact test, and you can get a particular idea for every transcription factor. But the problem is that you have multitude of transcription factors and multitudes of genes, right? It is not one transcription factor, but a many transcription factors, and it is cross-regulation. And for that, the problem is combinatorial. So the combinatorial problem is like this, that you have many different transcription factors and many different genes, and therefore you must do a permutation combination of all the transcription factors and all the different genes and ask whether in Fisher's exact test they stand together or not. The problem becomes computationally intractable because you very quickly go to something like 10 to the power 400 number of calculations that would need to be done in such a case. So you can't do 10 to the power 400 Fisher's exact tests. And therefore now you need to do some approximations. And that's exactly what I'm going to describe to you, what kind of approximations we make. And we show that you can still reproduce biologically relevant results. Now in any transcription regulatory network, typically what happens, for example, in this case I have taken the example of E. coli, is that it has about 200 transcription factors, about 4,000 genes. So about 200 transcription factors control expression of about all the 4,000 genes in E. coli. And some transcription factors control expression of larger number of genes. Some transcription factors control expression of fewer number of genes. Now that's to be expected. Things like, for example, global transcription factors such as HU, HNS, RPOS and all in E. coli would control expression of a very large number of genes. That is represented here. Well, certain transcription factors control expression of only 1 or 2 or 3 genes or something like that. So you have a natural distribution like this. Now, if you take any randomized network, suppose I were to come up with 200 transcription factors and 4,000 genes, and I assign directions randomly between these 200 and 4,000, what I would expect to get in a Fisher's exact test, I would get a uniform distribution like this. Unfortunately, what happens when you apply Fisher's exact test is you get a distribution something like this. And naturally, the transcription factors which have higher connections, they tend to be enriched in the Fisher's exact test. Now, that's a statistical flaw in the method, that transcription factors which have larger number of connections, they tend to be enriched in this case. And that's a statistical flaw. So we can apply certain corrections such as what is called as Bonferroni correction and all. But still, the problem remains that you always tend to enrich the transcription factors which have larger number of connections. So how do you address this problem? And very briefly, I will show you and then I will end here. So basically what we do is that we get an underrepresentation of transcription factors in the enriched list. That is, transcription factors with smaller target set size may not always be statistically enriched. On the other hand, the transcription factors which have larger connections, they statistically get enriched. Something like this. Alright. So what we did is we came up with something called a greedy search kind of an algorithm. It's a very basically go on adding transcription factors to an already identified one particular transcription factor and so on and so forth. So we tried 3 different greedy search algorithms. In the first step, we order all the transcription factors by their individual overlaps, what I showed you in the Venn diagram earlier, and calculating Fisher's exact test and calculating a p-value. And if the p-value is less than 0.05, we say that this list may be significant, and we order all of them by their p-values. Next, we actually perform 3 different methods. I will very briefly describe what the 3 different methods are, and we'll compare the 3 different methods. In the first method, we add the next TF in the ordered list and evaluate the combined p-value with the previous transcription factors. If the combined p-value is less than the previous one, we go on iterating this. And we iterate this until we actually get convergence. And when we get a convergence, we stop the iteration and we say that that's a optimal solution that we have observed. In the second method, we add the transcription factor from the remaining list with the best overlap p-value with the uncovered genes. The first transcription factor may cover a particular set of genes. These genes which are uncovered, we take transcription factors and test it only on the uncovered genes and evaluate a combined p-value. Once again, we iterate. When the iteration stops, we say that these many transcription factors give the best combined p-value. In the third method, we add transcription factor from the remaining set, which gives the best combined p-value, and once again iterate and like this. So basically, there are 3 very binary variants of each of the greedy search algorithm. And when we do that, we get actually very spectacular results. In the end, we get p-values of the order of something like -40, 10 to the power -40 or -50 that suggests a very significant solution to a problem, that for a given macroarray, we can actually say that these 20 transcription factors with a very high statistical significance control expression of all the genes, something like that. And I'll just show you very quickly. So the number of times that we get the converged solution in the third method that I showed you, right, actually generally significantly better than the first 2 methods. So we'll restrict ourselves only to the third method that I described to you in the previous 3 slides. Then method 3 seems to work actually better than other things. And what I've shown you here are the macroarrays performed under different environmental conditions compared to normal conditions. For example, at pH 5, pH 8.7, under norfloxacin stress, under sucrose stress, under sodium chloride stress, and so on and so forth. Right? So different macroarrays we have compared and come up with an optimal solution for the set of transcription factors that would do. And interestingly, how quickly you can actually arrive at a solution. the method C very quickly arrives at a solution with a p-value which would be of the order of -60 or -70, and any random number of tests that we do, in the minimum most steps, we method C actually arrives at a particular solution. So the last method that I described to you is one of the best one. I will just now describe one application of this to cancer microarray data, and I will stop there. So basically what we have a method now is to tell you from microarray data, we can tell you that what transcription factors are responsible for the observed microarray data, observed differential gene expression data, right? So this is an interesting problem, because now we can actually target only this small set of transcription factors instead of all the 1000 or 2000 genes that you actually had in your differential gene expression. So in this case, what we have done is we have taken a curated background network consisting of about 50,000 transcription factor gene interactions, which span about 300 transcription factors, right? I mean, these are the background network that we have taken, and we have taken certain cancer microarrays. For example, in this case, we have taken 4 ER negative cancer cell lines, which people have done the microarrays on them. So we have not done the experiment ourselves. We have picked it up from the literature, and 3 ER positive data sets, and the macroarrays which are available for all of them. We have actually run this method and try to arrive at the set of transcription factors that actually control expression of so many different genes. So basically what we have is that 6 ER positive versus 12 ER negative samples obtained from these experiments, which are already shown on the slide here. All right. And what we have done is that for the macroarrays, we have performed a SAM analysis so that the false discovery rate is less than 0.01, and log fold change is at least 1, something like this, or what you have taken. And we see that if we apply a single method, that the first ever method that I described to you, that applies single Fisher's test and find out which transcription factor it is, we actually come up with a list of transcription factors which is like this. And when we do combinations of different transcription factors, we arrive at these transcription factors which might be responsible for ER negative versus ER positive cancers. Right? In the microarrays that people have performed for the different cell lines for ER positive and ER negative, we say that these are the transcription factors that might be responsible for the observed microarrays. So the value of the method that we have proposed is in trying to identify transcription factors given differential gene expression. Now this is applicable to any network. As I said, it's a very generalized method. Any regulated network or directed network, we can take it and apply it to that. For example, if you have a network of kinases and their substrates, right? The transfer of phosphate is from a kinase to the substrate. That becomes a directed network. So genome-wide network of all the kinases and their substrates. Now once again we can actually go back to this. And if someone has done a phosphoproteomics data, we can apply this method and tell them that these are the kinases which were actually responsible for the observed phosphoproteomic data. So all the phosphorylated proteins, if you measure in a cell at any point of time, we can tell, depending on the network that what we have in the background, that these are the kinases responsible for the observed phosphorylation data. Something like this, right? So work back from? Yeah, exactly. So the idea right now, what we are working, and we are pretty close to that, is that given differential gene expression, you identify transcription factors. Given the transcription factors, now we have a set of about 25 transcription factors as shown here. 25 transcription factors, superimpose this on the phosphorylation network and identify which kinases. So that's the grand idea to actually do this in this case. Yeah. All right. So to summarize what I have told till now is that it is possible to make approximations and generate context dependent networks. So what I showed you in the first example is that take a microarray, map it on the network, remove the nodes which are not expressed. So what you have now is a context dependent network. Right, a network which represents only the genes which are expressed under that particular condition. And now you can start comparing many different networks which are context dependent. So under different contexts, you have different networks, you can compare them. Certain regulatory motifs appear to be enriched in nature. The reasons for this we don't know, but it's a very important philosophical problem and we need to prove it further. And it's possible to generate a comprehensive transcription regulatory network using differential gene expression data, is what actually I showed you in the last thing. And although I showed you transcription regulatory network, as I said, it can be generalized to many different regulatory networks. I mean, not only transcription regulatory network, but any regulatory network that you can think of, you can apply this particular problem method to that particular one. So to end, I would like to thank all my former and present colleagues. So Selu Yala Buna, Manimaran and Gaurang Mahajan, they have been postdocs in my lab. Kshama and Shubha have been my PhD students. I have collaborated extensively with Kanuri on the cell cycle network and with Sharmila on Boolean modeling of networks. And Seva Makiv, my collaborator in the Russian Academy of Sciences, we actually do a lot of these differential expressions and microarrays and all things together. I started this work about, as I said, 10 years ago. It was originally funded from the institute where I was earlier, that is the CDFD Hyderabad. In the last 5 years, I've been working at NCCS Pune, and a lot of support comes from the core funding of this. But I also get funding from DBT and DST. Thank you.